G'day, welcome to DNA Lives Forever, where I talk about various topics related to DNA, health, and/or biological immortality. I'm David A. Elliott, and this is the third and final episode where I am discussing genetic research into the deaths of Kathleen Folbigg's 4 children, for which she was sentenced in 2003 to a minimum of 25 years in prison and labelled as Australia's worst female serial killer. Recent genetic research indicates that at least 2 of the children most likely died from natural causes due to rare genetic mutations, which has resulted in her pardon and release from prison this year on the 5th of June 2023. In this third episode, I will focus on discussing how the most recent research may provide a natural explanation for the deaths of Caleb and Patrick. But before I do, I will briefly summarise what was covered in the first 2 episodes. Kathleen Folbigg had 4 children who died as infants from 1989 to 1999. At the time, the deaths appeared to be from natural causes. Investigations into possible genetic causes began after the death of her second child, Patrick, in 1991. At the time, this required determining which genes could be related to their deaths and testing for known mutations. No genetic cause was found, but it is possible mutations in other genes could have been responsible and left open the possibility of a genetic cause. In 2003, Kathleen Folbigg was convicted of killing her 4 children based on circumstantial evidence. The development of whole genome sequencing led to a further genetic investigation in 2019. This process, in effect, is the reverse of the process followed in the 1990s, with effectively all mutations in each child's DNA being found and investigating if each one's effects on the genes they occurred in could have caused the children's deaths. For Sarah and Laura, a mutation in the CALM2 gene was found to have a significant likelihood of causing sudden cardiac death, particularly in association with a modifier gene and respiratory infections that both had. And in the case of Laura, medication she was prescribed and her myocarditis, the inflammation of the heart muscle. This leaves Caleb and Patrick, and I will now discuss what the recent genetic investigations found for them. As I mentioned at the end of the second episode, there could be another gene involved here that the researchers did not include in their original analysis, but did mention in their supplementary material. This is called the BSN or Bassoon gene. Now, I had to do a bit of additional research, but this gene is mainly active in neurons, meaning it is important in the nerves and brain. And it seems it is a recessive gene. I'll just briefly and simply explain what a recessive gene is. Our genes come in pairs, with each one in the pair called an allele. We get one allele from each parent. Now, if one of them has a mutation and it doesn't work as it should, then for a recessive gene, it doesn't matter, as the other working allele can compensate for it, and everything is okay. But if both alleles are mutated and not working, this is when problems may arise. For the Bassoon gene, it was found that Kathleen Folbigg and all her children had the same mutation in one of the alleles in the gene pair. Future David here. I just have to interrupt as past David has gotten confused about who had what mutations, and I will just make some corrections. Kathleen Folbigg's mutation was only present in Caleb and Patrick, but as Kathleen's other allele in the Bassoon gene pair had no mutations, she was unaffected. However, all 4 children had a different mutation in one allele of the Bassoon gene pair that was not present in Kathleen. Consequently, this would have come from their father, Craig, but he did not provide a sample for testing, and that is why he is not shown as testing positive for it. Sarah and Laura's other allele, inherited from Kathleen, had no mutations, and so they were unaffected. As Caleb and Patrick had non-functioning Bassoon alleles from both parents, they did not have a working copy of the Bassoon gene, and problems could arise. And I'll let past David continue with discussing those problems. Back to you, past David. So, what are these problems that could arise? Well, the researchers' supplementary material shows that other research on mice with the same gene has found if they do not have a working copy of the Bassoon gene, that it results in early onset lethal epilepsy. Now, I read the other research paper they referenced for this. This stated that at birth the mice appeared normal and developed normally with no apparent physical abnormalities, including brain structure. However, the mice had a greater likelihood of frequent epileptic seizures, and more than 50% died within 6 months of birth from these seizures. Now, mice aren't people, so the effects of this gene could be different in humans. But does any of this have similarities to Caleb and Patrick that could explain their deaths? Caleb died at 19 days of age in 1989. He appeared normal at birth, but had difficulty breathing and transient tachypnea. This is abnormally rapid and shallow breathing at birth that usually resolves within a few days. He also had laryngomalacia, which is a birth defect in the voice box, called the larynx, where the soft tissues of the larynx fall over and partially blocks the airway. It was considered it may have been a factor in his death, but unlikely to have caused it, and his death was originally recorded as SIDS. There does not appear to be any apparent similarities between the mice and Caleb's death, but it may be possible he had an unobserved or unidentified seizure leading to his death, with his breathing difficulties being a factor, but this is unknown and his death is still considered as a SIDS case. Patrick died in 1991 at the age of 8 months. Everything appears to have been normal up until he was 4.5 months old, when he was found in his cot, pale and limp, with faint and laboured breathing, but responded to oxygen treatment. However, in hospital, seizures developed that did not respond to treatment. A CT scan of his brain showed abnormalities in the temporal and occipital lobes. He was discharged with a diagnosis of intractable seizures, probably viral encephalitis, that's inflammation of the brain, and bronchiolitis, a chest infection. Over the next 4 months leading up to his death, he had multiple admissions to hospital for epilepsy, gastroenteritis, and oculogyric crisis. This is where the eyeballs spasm into a locked position, usually upwards. He was diagnosed as having major epilepsy with cortical blindness. This type of blindness being due to damage to the brain's cortical lobe. The epilepsy was presumed to be caused by near asphyxiation at 4.5 months. He was found dead or possibly near death in his cot. Autopsy showed gliosis in parieto-occipital areas. This is basically a build-up of a type of cell in the brain after an injury to that part of the brain, but also occurs with such things as viral encephalitis, MS, Alzheimer's, and stroke. He also had hepatic congestion, which is basically an inflammation of the liver and is often caused by heart damage. It was assumed that both of these autopsy observations were due to his near-death event 4 months earlier. Consequently, Patrick's death was originally recorded as asphyxia due to airway obstruction and epileptic fits. There are some apparent similarities between the events that occurred with Patrick and the mice with the non-functioning Bassoon gene. The main similar points being born apparently normal, but developing frequent seizures and eventual death as a result. It appears the explanation at the time was that the near-death event at 4.5 months may have caused the seizures, but perhaps it was the other way around. It is possible that seizures resulting from the non-functioning Bassoon gene may have started with and caused the near-death event. Unfortunately, further research would need to be carried out to determine the exact effects of a non-functioning Bassoon gene in humans and how similar or dissimilar they may be to the mice from that study. At present, the probability that the Bassoon gene could have caused the death of Caleb and Patrick seem less clear than that from the CALM2 gene in Sarah and Laura's case. However, it is a possibility, and the probability may become clearer in future with further research into the Bassoon gene. For example, in December 2022, a research paper published in the Journal of Medical Genetics establishes mutations in the Bassoon gene are associated with epilepsy in humans. In any case, there is now much stronger and direct evidence that the children died of natural causes, with no direct evidence supporting an unnatural cause of any of the deaths. This has strong support from many eminent scientists and medical experts, 91 of whom endorsed the 2021 petition calling for Kathleen Folbigg to be pardoned based primarily on this most recent genetic investigation. The hearings of a new inquiry commenced on the 14th of November 2022 and concluded on the 27th of April 2023. At the conclusion of these hearings, the Department of Public Prosecutions conceded they now believe there is reasonable doubt as to the guilt of Kathleen Folbigg.
The Director of Public Prosecutions has accepted that new scientific evidence casts doubt on the mother's guilt.
That on the whole of the body of evidence before this inquiry, there is a reasonable doubt as to Ms Folbigg's guilt.
The Chief Justice, who presided over the inquiry, will now write his report and make recommendations. The writing of his report may take some time, and the process of actioning his recommendations could take longer. So, there were calls for the Attorney-General to recommend to the Governor-General that Kathleen Folbigg be immediately released through a pardon or early parole instead of leaving her in jail until the process is completed. After discussions with the Chief Justice for his views, the Governor-General pardoned Kathleen Folbigg on the 5th of June 2023, and she was released from prison on the same day. But I'll leave it there, which brings us to the end of this series of episodes on DNA Lives Forever. I hope you enjoyed learning about the genetic investigations into the cause of death of Kathleen Folbigg's 4 children and the doubts it has raised about her convictions for their deaths. If you liked this topic, make sure you follow DNA Lives Forever and hear me talk about other interesting topics related to DNA, health, and/or biological immortality.